If you can hear me, please inform me to start. Okay, very good. So 2 hours back, I have sent the file in WhatsApp group. Please go through it. You find it. I think there are many messages after file uploading you had. So 2 hours back, you can find it. Are you in Corynebacterium diphtheria? So another group, I think one lecture is Corynebacterium diphtheria, okay? So 67. Corynebacterium diphtheria. As its name shows, it is the causative agent for the disease of diphtheria. As you know, one of the most important functions of lysogeny. What is the meaning of lysogeny? Some viruses or some phages or some bacteriophages can send their nucleic acid, and viral nucleic acid can join. Viral nucleic acid can join to the bacterial chromosome, which we call it prophage. And the phenotype of toxin production is related to the genes of virus which incorporated into the bacterial chromosome. So as you see, one of the most important functions of lysogeny from a medical point of view is the synthesis of several exotoxins in bacteria, such as, so in some bacteria, such as the causative agents for diphtheria, botulinum, chlorella, erythrogenic toxins of Streptococcus pyogenes, all of this bacterial capability to produce toxin are related to the viral genome. So botulinum we explained, chlorella we explained. Now it is the time of explaining diphtheria, and after some times, maybe in the future, very soon, we go through the erythrogenic toxins of Streptococcus pyogenes also. So by this introduction, now we understood that Corynebacterium diphtheria, by help of its friend, which it is a virus, lysogenic virus, can produce toxin. So encoded by the genes of integrated bacteriophage, which we call prophage, toxin production is possible. Lysogenic conversion is the term applied to the new properties that a bacterium acquires as a result of expression of the integrated prophage genes. As you see, it is the schematic structure of diphtheria toxin, DT, diphtheria toxin. As this photo shows, it has two subunits, subunit A and subunit B. Subunit B contains two domains, R domain and T domain. B subunit is helping for attachment of the toxin, and the activity of the toxin is related to the subunit A. So it is the structure, or it is schematic structure of diphtheria toxin, or DT. So it is catalytic site. Catalytic site. Subunit A is catalytic site. Problem is related to the diphtheria, is related to this part, and R domain as receptor and T as transmembrane. They are working for attachment and helping for transfer. C domain inside the cytoplasm of human cells. Here you can see this is subunit, bigger subunit, or subunit B. Subunit B is composed of two parts. You can see 1, 2, and this is 1, 2, and this is A site. And as it is obvious by these sulfide bonds, B subunits and A subunits both can attach to each other, and they show this appearance. So it is diphtheria toxin. It is the diphtheria toxin. Okay, so what did we understand? We understand that those Corynebacterium diphtheria without phage are not so harmful, and very easily immune system remove them, suppress them, kill them. But those Corynebacterium diphtheria which lysogenic phage can send the gene inside the bacterial cytoplasm, and diphtheria toxin gene is the part of viral genes which incorporated to the bacterial chromosome. Now this prophage can help for toxin production. Beta phage carrying diphtheria toxin gene infects Corynebacterium diphtheria, not lysogenized by beta phage. Bacterium is non-pathogenic prior to infection by beta phage. So if there is no beta phage, there is no disease, there is no symptoms. Immune system kill this bacterium. But if there is lysogenized, if the genes of virus incorporate, now this bacterium has higher virulence. Now it is very pathogenic. It is very pathogenic. So beta phage genes are very important because they contains the diphtheria toxin gene. So beta phage and Corynebacterium diphtheria are good friends. Bacteria protect viral gene, help the genome of virus multiply, and viral gene is good friend for bacterium because help it to have toxin and accordingly elevated its virulence. Now it is more virulence. Diphtheria toxin genes integrated into the chromosome of Corynebacterium. Diphtheria bacterium becomes pathogenic. So these two bacterium completely are the same. The only difference is the prophage. This is containing prophage according lysogenic conversion, and this is without prophage. So it is not pathogenic, and this is severe pathogen. Severe pathogen. In this figure, what can we see? We can see a lysogenic conversion. Lysogenic conversion happened. In the left hand panel, in this part, left hand, transduction of the diphtheria toxin gene by beta bacteriophage results in lysogenic conversion of the non-lysogenized, non-pathogenic Corynebacterium diphtheria. So now Corynebacterium diphtheria receive the viral gene, beta gene. In the right, in this side, in the right hand panel, the recipient lysogenized bacterium can now produce diphtheria toxin and can cause the disease diphtheria. Note that no progeny phages are made. There is not uncontrolled multiplication of virus. When bacteria multiply to 2, one phage going to left and one going to right, and then 4, 8. So multiplication of bacterium and viral genome are in parallel and equal. They are in parallel and equal, and there is no harmful interaction coming from virus. Note that no progeny phages are made within the lysogenized bacterium because the diphtheria toxin gene has replaced some of the beta phage genes required for replication. The beta phage therefore cannot replicate. The lysogenized bacterium is not killed by the phage and can multiply, produce diphtheria toxin, and cause disease. It is why some Corynebacterium diphtheria are in less virulence and some are in high virulence and they are highly pathogenic. It is only according absence or presence of prophage of beta phage. 4 main steps involved in intoxication of a single eukaryotic cell by diphtheria toxin. First, we go to the photo to see what happened. Now, suppose, what is this? This is Corynebacterium diphtheria and produce toxin because this bacterium is lysogenized because this bacterium is lysogenized. homogenized. So what happened? Diphtheria toxin, as I told you, it has two parts, two subunits, A and B. B is responsible for attachment to what? To receptor, to toxin receptor. So toxin, according B subunit, attached to the receptor on the cells, receptor on the cells. And then what happened? Toxin enters through receptor mediated endocytosis. Now it is endocytosis. Endocytosis by the interaction of the key and lock. Key is B subunits and lock is the receptor. So the door is opened. This open door we call as endocytosis. You can see B subunits and A subunit, membrane, receptor mediated endocytosis. When this coming inside the cytoplasm of our cells, human cells, B remaining in phagosomes, but A catalytic site, active site, going out of the phagosome and going to the cytoplasm. Going to cytoplasm. Acidification of endocytic vesicle allows A to dissociate from B. Here acidification happens. And only A subunit can coming outside the phagosome. And now it is inside the cytoplasm. It is inside the cytoplasm. A enters cytoplasm. Okay. And going to find ribosome. And in ribosome going to find EF2. Going to find EF2. Don't forget we are talking about human ribosome. The toxin is going to our cells, cytoplasm. A subunit going to find ribosome. Then it find the enzyme elongation factor number 2, which is used for translocation. What is the translocation? When one amino acid comes to the ribosome, ribosome 3 codons should move. EF2 is used for movement of ribosome 3 codons each time on mRNA. 3 codons should go during elongation. So this is amino acids. Polypeptide is forming, and A subunit of acid find EF2 and stop it. So when EF2 is stopped, no movement of ribosome on mRNA is not possible. So it stops protein synthesis also. So it stops protein synthesis of what? Of our cells. So our cells cannot produce new polypeptide. You know, Protein synthesis is a vital action in the metabolism of cell. If one cell loses its protein synthesis, will die. So these toxins, these bacterial toxins kill. This bacterial toxin kills our cells by stop working EF2 and stop translocation of ribosome on mRNA and stopping protein synthesis. Okay, so it is the mechanism of coming the toxin inside the cell and doing some negative behavior. It is what I explained. Don't confuse it, only we read it because it is very simple. Yes, finding key and lock. So by attachment of toxin by B subunit to receptor, endocytosis start, then acidification of phagosome, A subunit can come out, B subunit again return back to outside the cell, A subunit attack to EF2, and then there is no translocation and there is no protein synthesis in our body's cells. Okay, it can kill our cells, and it is what we told. One, the beginning of the toxin to the surface receptor of its target cell. Okay, grouping of charge receptors into layered pits and internalization of the toxin by receptor-mediated endocytosis, okay, followed by acidification of endocytic vesicle by a membrane-associated ATP-driven proton pump. So acidification is done by a membrane-associated ATP-driven proton pump. 3. The insertion of transmembrane domain B subunit into A subunit to the cytosol and the ADP ribosylation of elongation factor 2 or EF2, which results in the permanent inhibition of protein synthesis as shown in the next page. A single molecule of catalytic domain delivered to cytosol is enough to be deadly for the cell. A single molecule of A subunit is sufficient to kill one cell. Only one molecule is sufficient. During translation, EF2 mediates the translocation of mRNA. Here is explaining what is the importance of EF2 and why if EF2 not working, cell go to die. During translation, EF2 mediates the translocation of the mRNA relative to the ribosome, enabling the codon shift from amino acyl A site to peptidyl P site and finally to exit site in a GTP dependent reaction. If you forget it, if you forget initiation in protein synthesis, elongation in protein synthesis, and termination in protein synthesis, very simply you can go through the internet or book and review it. 100% all BST students know it very well. Know what? Know the proteins, mechanism of protein synthesis. So protein synthesis has three parts: initiation or start with IF1, initiation factor 1, initiation factor 2, initiation factor 3. Then it continues in second part for amino acid polymerization. We call it elongation. And it is done by different elongation factor, like elongation factor 1 and 2 and the others. So it is what happened by Corynebacterium diphtheria toxin. It is the toxin. This is the microbial toxin and its mode of action, mechanism of action. Here again we can see in a better way what happened. Again we review it. If Corynebacterium diphtheria can go inside our body, which contain lysogenic phage, beta phage, so this bacteria produce toxin. This toxin has two subunits, A subunit and B subunit. We call it diphtheria toxin. By B site can attach to the receptor of cytoplasmic membrane of human cells. B component of the toxin attaches to host cell receptor, okay? Then only A is released in the cytoplasm. A component enters to host cytoplasm, inactivates elongation factor 2, prevents protein synthesis by ribosome, and kill human cell. One molecule is sufficient to kill one cell. Now we see in more detail what happened. Diphtheria toxin has two subunits, A subunit and B subunit. By help of B subunits, this toxin can attach to the toxin receptor. Diphtheria toxin's receptor binding domain, or B, binds host membrane. Now it is attachment. Not attachment of bacteria to the receptor. It is attachment of bacterial toxin, diphtheria toxin, to the receptor. Then what happened? Number 2, membrane-bound toxin, AB, enters by the endocytosis. Now it is endocytosis by cytoplasmic membrane and with induction by receptor-mediated endocytosis. Number 3, catalytic subunit A, this is catalytic subunit A, is cleaved from B site, B subunit, by the disulfide bonds, by dissociation disulfide bonds. The disulfide bonds are reduced after reduction of this disulfide bonds. Now A searching to find the ribosome and to find elongation factor number 2. The transmembrane domains facilitate passage of the catalytic A peptide through the vesical membrane. So catalytic site help A coming to the cytoplasm. catalytic A now. The catalytic A domain ADP ribosylates elongation factor 2 going and changing EF2 in such a way this elongation factor now halts protein synthesis and kills the cell. So when EF2 not working, there is no translocation, there is no polymerization continues. Polymerization not to be continued and cell go to die. Cell go to die. In brief for last time, very fast, tox bacteriophage give beta toxin gene to the Corynebacterium diphtheria. It produces toxin with A and B. By B, they attach to receptor. Then phagosome is sending them inside. Then by proton pump acidification, and only A is removed, is released, and A go find EF2, and by ADP ribosylation stop its job by stopping EF2. Finished. No protein synthesis. Without protein synthesis, cell go to die. Okay? Is it clear? Do you have any question? I think 3 times I explain it. Okay, very good. Now, as much as we have time, we go through new toxin. There is one bacterium which you know it, and its name is Staphylococcus aureus. Staphylococcus aureus. It is an important pathogen of human and livestock. It is pathogen of human and some animals. It causes a diverse array of disease, ranging from relatively harmless. Sometimes problem is very small, and it is in a short part of skin. But sometimes it can be dangerous, and sometimes it can be life-threatening systemic conditions. So from a very small simple disease to a very big problematic disorder may show itself. Among multiple virulence factors, Staphylococci secrete several exotoxins directly associated with particular disease syndromes. For example, some can produce toxic shock syndrome toxin 1 or TSST1. Some can produce enterotoxins, which is effective on our intestine and alimentary canal, and it is coming mostly by food poisoning. And the other one, the last one, which today we want to focus, and we discuss only this toxin, is exfoliative toxin. Exfoliative toxin, ET. Exfoliative toxin. So number 1 and 2 we will discuss later on. Today we want to focus on exfoliative toxin. Okay, what is it? It is particularly interesting as the sole agent responsible for staphylococcal scaled skin syndrome, SSSS, 4S, staphylococcal scaled skin syndrome, or writer's disease. What is this? It is a disease predominantly affecting infants and characterized by loss of superficial skin layer, dehydration, and secondary infection. So this infection is done by attack of some strains of Staphylococcus aureus to skin, mostly infants and children. Why rarely in adults? Because we have a strong immune system. Normally, Staphylococcus aureus cannot like it, but cannot attack to our skin because we have normal and strong immune system. But in infants and children, immune system is very weak. On the other hand, not all of Staphylococcus aureus. Some strains have the ability for production of these types of toxin. So it is our certain proteases. So these toxins are working like enzyme protease, which attack to the amino acids, proteinaceous structures with high substrate specificity, which selectively recognize and hydrolyze desmosomal proteins in the skin. So, number 1, we never forget that exfoliative toxin attack to desmosomal proteins in the skin. Proteins, yes, and this is protease. So protease break proteins. Proteins of what? Desmosomal proteins in the skin. When they do that, they are responsible for staphylococcal scaled skin syndrome. Sometimes this illness or this syndrome is localized, only one part of the body. Sometimes it is whole body, in the face, in the chest, in the chest, in the back, in the hand, on the hands, on the foot, on the feet. Whole body can go over the surface of the body. So again, we remind you, some strains of Staphylococcus aureus can produce this disease if they can produce exfoliative toxin. If they can produce exfoliative toxin, it's caused by exfoliative toxin, and they are 2 types, type A and type B. Exfoliative toxin A, ETA, exfoliative toxin B, ETB, of Staphylococcus aureus. These 2 types of toxins have many similarity and a few differences. So they are very similar in action and a few differences. Early staphylococcal scaled skin syndrome, or 4S, manifest or start with fever, malaise, lethargy, and poor feeding. Don't forget, we are talking about children. Don't forget, we are talking about infants. That after some time, it followed by erythematous. You can see a red appearance. Erythematous rash. And this rash join together and take a surface of the body. If it is on one part, it is localized. And sometimes it can be all over the skins of the body. And the formation of large, fragile, fluid-filled blisters. So sometimes you can see over here, they can be bigger, and under the skin there are some pus and liquid, like here you can see it. They can have like this with pus and some liquid. The blisters burst with mechanical action, leaving the affected parts of the body without the protective layer of epidermis. Don't forget that our epiderm on the upper side of skin is the first protective barrier in front of bacteria. Okay? So now all our tissues are sterile because epidermis don't allow bacteria going down to the deep tissues. Okay, and this bacteria open this gate. This bacterial toxin make our epidermis like a gate and allow all bacteria go to our tissue and make big problems to infants and children. So the blister burst with mechanical action. So by itching, by rubbing, by finger, by nail, infants and children push and take on skin. Mechanical action, leaving the affected parts of the body without a protective layer of epidermis. Only the skin, but not mucosa, is involved in this situation. Severity of disease varies. A few blisters localized to the site of infection, but in severe exfoliation affecting almost the entire of body is involved. So sometimes a few blisters in one place, sometimes all over the body involved with the problem. A localized form of 4S restricted to the site of infection. Here it is restricted. Yes, it is only restricted to one part of the face. A localized form of 4S restricted to the sites of infection is recognized as bullous impetigo. Both conditions share the same etiology and differ only in the extent of skin damage. So as I explained and you understood, we have many exfoliative toxins, which two of them are very important in human infections. Exfoliative toxin A ETA and exfoliative toxin B, ETB. The ETA and ETB serotypes are homologous, have molecular masses of approximately ETA 27 kilodalton and ET and the other one, masses of 27 kilodaltons both, and the number of amino acids are 242 in ETA and 246 in ETB amino acids. They are protein. These toxins are composed of amino acids. The ETA is heat stable, heat resistant, whereas ATB is heat labile or heat sensitive. Both of them distinguished by the extent of damage caused in the epidermis. Both are the same infection, but one is milder and the other is severe. Forest occurs mainly in newborns and children, with outbreaks in adults being rare. I explained why. Because adults have normal immune system, but children immune system is immature. The greater susceptibility of children has been attributed to the immature immune system, weak renal clearance of the toxin. In children, kidney cannot very easily and fast clean the body, clean the liquids of the body like blood from this toxin. But in adults, it is done soon. And the fact that children are common carriers of the microorganisms. Approximately 5% of all Staphylococcus aureus strains produce exfoliative toxins. So it is not so, which we think all staph can do it. Only 5% have the genes related to these toxins, with ETA being most prevalent in Europe, Africa, and America, and ATB being more common in Japan. ETA is confined by the ETA gene. So ETA is a phenotype. It is a toxin. This toxin is the phenotype, and it has gene, and its gene is ETA, and it is located on chromosomal DNA, carried on genome on a temperate phage, but not related to the chromosome. It is related to the temperate phage, like before, as I explained. So only those one with prophage, with temperate phage, can produce ETA. So now we understand why 5%, because some doesn't have this friend. Some of the Corynebacterium don't contain the prophage, but ETB as a phenotype is related to ETB gene, and it is not related to virus and not related to chromosome. It is related to the large plasmid DNA. It is related to large plasmid DNA. The ETB plasmid has multiple genes, but this plasmid doesn't contain only the gene related to toxin. It contains many other genes, especially those genes which they are related to antibiotic resistance, which contributes to the increased resistance of ETB positive Staphylococcus aureus strains. So those bacteria which have toxin, those Staphylococcus aureus which contains ETB is a virulence factor. On the other hand, they have antibiotic resistance in their plasmid, and it is another virulence factor. So not only they can produce toxin and invade to our cells in our body, but also when we use antibiotic, they are resistant to most of them. They are resistant to most of them. ETA toxin was found to be associated with bolus impetigo. As I explained you, here you can see bolus impetigo, Staphylococcus aureus produce toxin, cleaves superficial skin layer, and you can see there are some bolus containing clear yellow fluid full of pus, and when these are broken, many other bacteria can go through this infectious part and can be mixed infection with staph and another bacteria. Clinically, mostly it is common for kids, and sometimes in adults. Consider HIV. As you know, those people which are infected with HIV, human immunodeficiency virus, or those persons which have AIDS, HIV make the immune system weak and sometimes stop the immune system of person. So as I explained you in advanced bacteriology, some pathogens are true pathogens and some other pathogens are opportunistic pathogens. What are the opportunistic pathogens? Opportunistic pathogens cannot attack and start disease in people with normal immune system. And now here, this Staphylococcus aureus is opportunistic pathogen, waiting to find children, waiting to find infants with weak immune system, waiting to find people with containing HIV with weak immune system, waiting to find those people receiving kidney, those people which their kidneys cannot work properly, they should receive new kidney from another person. So physician give them immunosuppressive drugs. Okay, so these drugs make their immune systems weak, so they are prone to infected with this opportunistic pathogen. Okay, and we don't talk about therapy because we want to focus only on the toxin. We don't talk about epidemiology. We don't talk about the treatment because our lecture, our course is related to microbial toxins. We don't want to go through another aspects. because we have limited time to finish not all but most of bacterial toxins. So it is ETA was associated with bolus impetigo. ETB toxin was found to be associated with staphylococcal scaled skin syndrome, as you see, like this, with rare bolus, and mostly skin is in this fashion, staphylococcal scaled skin syndrome. So what happens? What happened? These toxins or exfoliative toxin have been described and are exotoxins produced by certain Staphylococcus aureus strains in the epidermis. You can see here is epidermis, basement of the membrane, and dermis. Now it is the place of action of this toxin. This bacteria, by help of its toxin, like to remove this part. Then what happened? It is like this. Yes? He wants to remove epidermis. This toxin wants to remove the epidermis, wants to separation of epidermis and dermis, blister formation. So now it is blister formation. What is the mechanism of action? What is the mechanism of action? As you see in this photo, these epidermal cells, cell in the right and cell in the left, they are connected together. Some structures are inside right cell, some structures are inside left cell, and then by these structures, these cells are connected. It is what now it is in our cells. Now all of us epidermis is okay because this interaction happens, this bond is happen. It is desmosome adhesion. What it is? What is it? It is desmosome adhesion. Okay, so it is normal case. What the toxin wants to do? Toxin wants to break it. Toxin wants cells of epidermis not connected to each other and remove very easily desmosomal cadherin type adhesion molecule or pemphigus foliaceus autoantigen is the target of Staphylococcus aureus exfoliative toxin. So exfoliative toxin action is this side here in between the connection of two cells. The exfoliative toxin, also known as epidermolytic toxin. Yes, it is a toxin and wants to lyse the epiderm. Wants to lyse the epiderm by breaking the bond, the connection between epidermal cells. They are certain protease because these are proteins. This toxin is protease. Wants to break proteins. They are certain proteases secreted by Staphylococcus aureus that recognize and hydrolyze desmosome protein in the skin. So desmosome proteins are proteins and toxin is protease and wants to break this point, and cells of epidermis not connected to each other. Diagram of desmosal adhesion. The key molecular components are shown that allow desmosomes to tightly link to adjacent cells. It is normal case. It is the normal case. Now after infection, now we want to see the mechanism of action after toxin action. Mechanisms of exfoliative toxin associated with keratinocyte dissociation. As you see, desmosome we show you in two colors, red color and blue color. Red color and blue color. Some things are inside the cell, in the right cell, some things are in the left cell, and some things, desmosomes are in between left and right cells. Okay? So it is exfoliative toxin. Exfoliative toxin attack to DSG1, the red ones. You can see there are 4 red ones, 2 related to the left cell and 2 related to the right cell. And exfoliative toxin break the red ones. When the red ones are broken, the blue ones don't have the power for remaining, for continuing this bond, and they are broken also. So what happened? There is no bond, there is no connection between epidermal cells. There is no connection between epidermal cells. Desmosomes composed of 2 major transmembrane Catherine type adhesion molecules, 2 molecules, red and blue. One is DSG and the other is DSC. DSG and DSC. DSG is red, DSC is blue. And cytoplasmic plaque protein. These proteins are inside right cell and inside left cell. We call them cytoplasmic plaque proteins that links between desmosomal, Catherine, and intracellular cytoskeleton. Figure possible mechanism of exfoliative toxin induced keratinocyte dissociation. PG. What is PG? It is PG, yes. Here, this green one in the cytoplasm. Internal skeleton, okay? PG. PG, plakoglobulin. Cleavage of the extracellular segment of DSG1, the red one, extracellular segment DSG1, initiate keratinocyte suppression, sequestering of PG by truncate DSG1, and disintegrity of other desmosomal components are associated with expansion of intracellular space between keratinocytes. So keratinocytes in the right and left, now they don't have any interaction. They don't have any bond. Here again you can see it is another photo. It is the upper side of skin is stratum corneum, stratum granulosum, stratum spinosum, stratum basal. Okay, and the most important thing for connection is DSG1. These blue ones are very important. DSG1. Figure different distribution of desmoglein isoforms in the epidermis explains the exfoliative toxin-induced splitting at the stratum granulosum. Mode of action is stratum granulosum. Schematic representation of desmoglein distribution in A, it is normal case. In A, you can see normal case in healthy skin, and B, skin exposed to exfoliative toxin affected, and the bonds between stratum granulosum cells are disrupted. Skin exposed exfoliative toxin. In all strata except stratum granulosum, the exfoliative toxin mediated hydrolyzed by desmoglein 1 is compensated by desmoglein 3, and desmoglein 3 is absent in stratum granulosum, which explains the cell detachment and splitting of the epidermal layer upon the hydrolysis of DSG1. So here you can see now it is a space between cells, and very easily the stratum, this one, the stratum corneum and the stratum granulosum are removed, and bacteria very easily can come to the deeper parts of the skin, So again, at last, we want to say, how does exfoliative toxin producing Staphylococcus aureus penetrates epidermal cells? Through firm keratinocyte adhesion in the upper stratum granulosum. How bacteria can go there and start producing toxin? Number one, Staphylococcus aureus adhere to the living epidermis. You can see like this. 2. Neutrophilic recruitment to the epidermis. Neutrophils come to defend. These are neutrophils are coming. 3. Penetration of Staphylococcus aureus through neutrophil created intracellular gap. When neutrophils are coming, some gaps happening between some cells, and this gap is a good way for bacteria to go to the deeper part and start producing toxin. Expansion of blister by exfoliative toxin cleaves DSG1 in superficial epidermis, and so you can see there is a big gap between upper side and deeper side of the skin. Figure bacterial cutaneous invasion and blistering in bolus impetigo. Staphylococcus aureus harboring at gene adhere to the living epidermis recruits neutrophils. When neutrophils create intracellular gap between superficial keratinocytes, Staphylococcus aureus produce exfoliative toxin to expand the blister by cleavage DSG1. And this is the last photo. As you see, what happened? These are Staphylococcus aureus. Staphylococcus aureus adhere to the epidermis and then penetrates through neutrophil created intracellular gap. This gap is created by neutrophil, and bacteria can come inside according to this gap between superficial keratinocytes. Expansion of blisters by exfoliative toxin, now destroying and removing the connection between cells and now it is proposed model for bacterial invasion and blistering provoked by staphylococcal exfoliative toxin. So our lecture today finished. If there is any question, you can ask. If not, we can finish and meeting each other next week. Okay, thank you very much. Hope the best. See you. Goodbye.